are not putting any requirements for permission to see any recordings. It's just a glitch in the program. We'll have to report it. They'll have to fix it. Just keep trying. Eventually, you'll be able to access this. But that's not anything we're doing, so it's not anything we can fix except for reporting a problem. Also, there have been questions about the grade to date. So over here on the far left, I have the grade scale to date. This does not include the lab quiz this afternoon. And then again, you want to go with percentages. Now, it's a little bit difficult to do percentages with Canvas because for me to have the gradebook include the extra credit points, I have to include it into the total. So what we have right now is a possible 460 points. And then those percentages in blue will make sense. So all your points in Canvas should be correct, but out of how many points possible, it's out of 460. So that's what the percentage is. And then there's your grade scale right now as we get close to the end. All right, are there any other housekeeping questions before we move on? I'm going to finish digestive physiology today and just kind of barely get started on urinary. I want to get at least through the anatomy review for urinary. The urinary system is one of the organ systems of our body that it's impossible to discuss the anatomy without the physiology and vice versa. So I'm going to make sure we're all up to snuff on the anatomy. Last time on the digestive system, I ended in the small intestine. So I had an outline on the whiteboard. And just to remind you of the colors, purple is mechanical processing, purple. Blue is secretion, things that our bodies put into the lumen or the donut hole, as I call it. Red has to do with the digestion. Remember, the word digestion means all the catabolic chemical reactions that are reducing these chains and the links. And then in green is the absorption of the links, plus some other things as well. I'll move the camera up so you can see everything. But I wanted to get this close enough that you could see the grade scale and the percentages, as well as setting up the rest of the whiteboard. Again, we left off in the intestines, and we were get a little closer. We're talking about how the most absorption occurs in the small intestine. And I only introduced this topic, and now we need to complete this topic because this is really crucial about where most of the absorption of nutrients occurs, and that's in the small intestine. So again, just continue on the whiteboard from the left bank and up on the top of small intestine where the most absorption happens. So there are three main reasons why the most absorption happens in the small intestine. We covered the first two. One was the location of the hepatopancreatic ampulla. I just put pancreas and bile. But remember that the pancreas and bile are introduced, the pancreatic enzymes, and the bile that's useful for fat absorption, digestion, and absorption, is introduced into the lumen at the very beginning of 20 feet of small intestine. So right there, you got the pyloric sphincter, the border between stomach and small intestine. 2 inches away, you got the hepatopancreatic ampulla. That's what the HPA stands for, hepatopancreatic ampulla. And that's where both the bile and the pancreatic exocrine enzymes are coming in, which was also written in blue. There, at the very beginning. So that's the first green point there. Make sure you catch it. Also, you have a controlled release of chyme at the pyloric sphincter. That was on the very last slides that we talked about. And then the first or second slide of the week 15 PowerPoint is the third point, and that's the 3 levels of surface area. And so on slide 2, it talks about these 3 levels. First, an overall understanding of what this surface area is doing, all these 3 levels of structures, is we have 70% of the donut hole is the small intestine. It's 20 feet approximately. Yet it has 90% of the surface area, and it's because of these 3 levels of surface area. So it has way more surface area than length of the digestive tract. First one are circular folds, or plicae circularis, and that is visible with the naked eye. If you take a magnifying glass to one of these visible ridges, you'll see it's covered with bumps, but you do need a magnifying glass to see that these bumps are intestinal villi. And then you've got to look at the apical surface of the simple columnar epithelial cell. And so if this is, you kind of see it here, the gut lumen right here, apical surface is what is up against the food. And that is covered with microvilli. And that really increases a lot of surface area, the microvilli. And so a lot of these pictures are showing you how intense this microvilli can be. And if you look at that one electron micrograph where it says microvilli of the brush border, if you imagine that top of that cell was flat right there about halfway through that picture, how much less surface area it would have than when it has the microvilli. So 3 levels surface area give this 90% of the surface area of the GI tract, even though the overall length of the small intestine is only 70%. Okay? All right. Then we have to talk about the absorption mechanisms themselves. If we get to those figures. So this is slide 4, and there's a table. And for the 3 nutrients, remember there are 4 biologically important macromolecules, but we don't count nucleic acids as one of the nutrients because you don't need to eat them. Your digestive tract will digest them and absorb nucleotides, but you really don't need to eat them. You can make all the nucleic acids that you need. None of the nucleic acids are essential. Remember, the word essential for a nutrient is something that your body can't make, so you have to eat it. So we only have to worry about carbohydrates, proteins, and lipids. And this is where you want to look at this figure on the far right that I drew. Okay, so simple columnar epithelium is guarding the gut lumen. And hopefully you can see this in the picture. There's 4 numbers, 1, 2, 3, and 4. That is the 4 columns of this table on slide 4. The absorbed form is what's in the gut lumen after it's all done with the digestion. When we talk about what's here, number 1, that is the single link form. 2 is how does it get in to the simple columnar epithelial cell. 3, how does it get out of the simple columnar epithelial cell? 4, does it head toward a blood capillary or a lacteal, the lymphatic capillary in the intestinal villus? So that's the 4 columns. Absorbed form is number 1, entry into microvilli is number 2, exit to vessels, that's number 3. How does it get out of the simple columnar epithelial cell? And then 4, does it go into a blood capillary or lacteal? This is, I have used this as a possible essay on lecture exam 4. So you should know it backwards and forwards. And this is a gross oversimplification. So, but let's just go with the easier form. All right, carbohydrates, the single link is going to be monosaccharides as a single link in the gut lumen after all the digestive enzymes has broken them all completely into single links. It has to be actively transported into the simple columnar epithelial cell. This is actually secondary active transport, but it's still active transport. Just put active transport, that's fine. Then when the concentration of these monosaccharides gets pretty high inside that simple columnar epithelial cell, the concentration gradient is driving it. It just has to go through a monosaccharide channel, number 3, in facilitated diffusion to get out of the simple columnar epithelial cell, and it's headed for the blood capillary. Boom. First row done. Proteins. Of course, our amino acids are the single links. It also is going into the simple columnar epithelial cell by active transport. Also, most of this is secondary active transport as well. But just put active transport. When the concentration gets high inside the cell, amino acids are small enough to go through the lipid bilayer. Single word, diffusion, out. And it's also going into the blood capillary, number 4. Okay, this is the bottom row here with lipids. We'll look at some pictures of these in the PowerPoint in just a second. But micelles are the smallest form of lipid bubbles after lipase has digested it. That's in the gut. That's the name of the tiny, tiny bubbles. They're very difficult to see. They're almost just little biochemical bubbles. And again, it's because the chyme is hydrophilic and fats are hydrophobic. Now, when we were doing lab 3, and I was talking about in the back table in 3C how pancreatic lipase breaks down triglycerides into glycerol and 3 fatty acids, that was an oversimplification. That's not even true. So I'm going to give you a little bit more of the actual accurate details. Lipase cannot break down triglycerides into all the way down to glycerol and 3 fatty acids. It can only break off the outside. If there's 3 fatty acids on a glycerol, lipase can only take off the outer fatty acids. It could take this outer one and it take that outer one. Everybody understand what I'm talking about here? So that's why what is actually number 1 for lipids is 2 fatty acids and a monoglyceride, because you cannot take off all 3. Lipase can't take off all 3. So that's number 1, the absorbed form. You guys like how I can work that in so I can flip off the whole class? Okay. Well, because it's lipid soluble, fat soluble, it can go right through the lipid bilayer. So it goes into the simple columnar epithelial cell by simple diffusion. Then here's the bizarre part. Once it's in that cell, this cell will put it back together as a triglyceride. Then it's going to put a whole bunch of triglycerides in its own bubble called a chylomicron. Then it's going to spit out this bubble by exocytosis. So number 3 for this last row is exocytosis of chylomicrons. And it's going to bypass the capillary and it's going to go into the lacteal. Now again, this is an oversimplification. The vast majority of absorbed lipids goes into the lacteal, not all of them. But those lipids that go into the lacteal will not be filtered by the liver before going into this general circulation. So we were talking about this with the lymphatic system. it goes straight into the left subclavian vein by the thoracic duct without ever seeing the liver first. Okay, so the next few slides are just pictures showing you information that's in that table. Slide 5 is about polysaccharides, in this case starch, being broken down by by amylase into disaccharides. And then the following slide is showing you the breaking down into single-link glucose. You can see that the sodium that's in the lumen of the gut, this is slide 6, I believe. Let me just check. Excuse me, the table's on slide 4, and this figure with the single-link glucose being absorbed is slide 5. You can see the sodium is showing you that that's secondary active transport. If you guys go, you got to go back to lecture exam 1 to review secondary active transport. And then it's going to go out by facilitated diffusion. That's that purple ball is supposed to be a protein channel. And it's going into the blood capillary. Next slide. Is showing you the breaking down of protein. Large chains are being broken down by pepsin into smaller peptides. They're not single links. Remember that pepsin doesn't break down into single links. You need the trypsin and chymotrypsin and all those enzymes to break it down into single links. Let me just make sure I'm not losing count here. Slide 7 is showing you the micelles, the little small bubbles, because again, it is hydrophobic and it's in chyme, which is hydrophilic. So it's got to be these little bubbles called micelles. That's how it gets broken down into. Now, notice how it's 2 fatty acids and a monoglyceride. The one carbon that on glycerol that still has a fatty acid is the middle one. But that's how it goes in just by simple diffusion into the cell. And then in slide 8, it's showing you how that simple columnar epithelial cell repackages it into a triglyceride, puts it into a chylomicron, and then spits out the chylomicron by exocytosis 3, and it bypasses the blood capillary and goes into the lacteal. Slide 8. Slide 9 is just a review with other figures that I found on the internet. Again, a lot of this is pretty basic stuff. So when the first row on slide 9 comes up, that first figure is showing you how the sugars come in. Notice again, what I have in the table is an oversimplification that is not completely accurate. You can see that fructose will go in by facilitated diffusion. Let's just go with the table for lecture exam 4. Then when the second row shows up, you'll see the next figure, and that's showing you amino acids going in by active transport. Again, it is secondary active transport, and then going out by simple diffusion to the capillary. And then when the third row comes up, you can see the micelles getting broken down into monoglycerides and 2 fatty acids. It goes by simple diffusion in. It gets reconstituted by the simple columnar epithelial cell into triglyceride, put in a chylomicron that gets spit out and then put into the lacteal. But also notice on the top portion of that figure how short chain fatty acids will go in the blood capillary. So let's just go with the table for lecture exam 4, but it is an oversimplification. Okay, now let's talk about the large intestine. That's on the next slide. And again, once the chyme passes the ileocecal valve, the food changes names from chyme to feces. And remember that the colon is the large intestine, and the colon is the last part of the donut hole. The large intestine ends at the anus. And a lot of students get confused because they're going, well, you got the rectum. Yeah, that's part of the large intestine. Well, you got the anal canal. Yeah, well, that's part of the rectum. This is the terminal part of the GI tract. Even though it has more specific names at the very end, it's still large intestine. And it's doing peristalsis. We'll talk about defecation here in a little bit. Again, there's no digestion that's occurring in the large intestine because that was completed in the small intestine. So right here, red is digestion. It's done in the small intestine. There's no digestion down here in the large intestine. The only things that are absorbed here, and most of the nutrients are absorbed in the small intestine, but in the large intestine, it is absorbing salts. And because of osmosis, water follows it. It is basically conserving, because remember, in the blue, we put a lot of juice into the digestive tract. This is where we're getting a lot of juice out of it. Now, a lot of the important things that went into it stay there because they're doing their job, but it's still going in with a lot of salt and water. And you would have a difficult time maintaining your body fluid levels unless you pull that out. And that's what the large intestine is doing. What it's doing while it's pulling salts and water, that's also compaction. And it's compacting the feces as it's going ring around the rosy, ascending colon, transverse colon, descending colon, sigmoid colon, and out. You're going from having the runs to having normal feces, and your body is conserving fluids when it's doing that. Also, the other thing that it is absorbing, again, most of the absorption is small intestine, not all, but most of it. Very little in the stomach and very little in the large intestine. One of the things that is being absorbed there are vitamins, and that's because a lot of these vitamins come from your gut bacteria. Okay, so they're only made there. And there's this interesting little figure that covers the bottom right of the slide. What number is this? 10. And I want you to take a look at these numbers. If you didn't pay attention to math when you were in school, then this might be lost on you. But that's why you're supposed to pay attention in school. It says there in the stomach, 10 to the third power per ml. Those are colony-forming units, or basically would be bacterial count. Now you're talking about a cubic millimeter. I mean, we're talking about tiny is a milliliter. And that milliliter in the stomach with pH 1 to 2 has 1,000 colony-forming units per ml. Then you look at the small intestine, and it has 10 to the 4. The 10 to the 6 per ml. That is 10,000 to 1 million bacteria in a milliliter. It's tiny, tiny volume. Now look at the large intestine. And that is the number of bacteria in the large intestine. And that's why I don't care if you think you're a princess, that's why your poop stinks, because it's coming from this amount of bacteria, which is huge. That's why you have to have Peyer's patches in the end of the small intestine in the ileum, because these bugs want to go upstream, and you got to have Peyer's patches to hold it down so that the numbers stay more like this in the small intestine. But these bacteria is not just giving you your stink, it's also making some vitamins. Okay, slide 11 is talking about the fecal matter that's getting compacted all the way around, and it's talking about some of the chemicals those bacteria are putting into the feces that's giving it its stink. It's interesting, you know, when we have the cadavers in the cadaver program, whenever they get to the point of cutting open the large intestine, people are surprised, hey, you know, because there's feces in the large intestine and the cadaver, and it doesn't stink. Why? Because all that embalming stuff killed all the bacteria. It's the bacteria that gives it a stink. That's what it's saying in the first bullet. And then we're talking about defecation here at the end on slide 11. Again, it is very similar to vomiting. You do have to close airways and contract the diaphragm as well as the abdominal muscles, increasing the intra-abdominal pressure. And again, the sigmoid colon is in the abdominal pelvic cavity, and the very beginning of the rectum is still there. But then the rectum disappears out of the pelvic floor, and this pressure is putting pressure on the sigmoid flexure just before it goes straight to the rectum, and that helps to assist the last terminal peristalsis. Defecation is the terminal peristaltic movements of the tunica muscularis, and the very end of the tunica muscularis is the internal anal sphincter. Okay? And yeah, as it says on the bottom, I don't got any animations of this, you sickos. All right, so that is talking about the digestive tract. Remember, I introduced this topic by saying we tend to talk about this first, the digestive tract, alimentary canal, donut hole, and then talk about the accessory glands. We've already covered salivary glands and pancreas, and so the only other organs left are liver and gallbladder. All right. I think I've said this to you before, but I want to repeat it. The liver is a top tier organ, and you only have two in your body, your liver and your brain. There's no such thing, I mean, there are things as a heart transplant. There's no such thing as a brain transplant. Yeah, there is such a thing as a heart-lung transplant. There's no such thing as a liver machine that can keep you alive while you're waiting for a liver transplant. And that's why the brain and the liver are two top-tier organs, even above your heart and lungs. Your brain dies, you die. Your liver dies before you can get a transplant, you die. There is no machine they can hook you up to keep you alive while you're waiting for a liver transplant, okay? Top tier organ. It's involved in many organ systems. It's just mentioning some of its functions here on this slide 12. Metabolic detoxification. It's also working with the red blood cell cycle. It mentions that there in this first slide. Osmotic balance, it makes albumin. There's just so many functions. Metabolic is that when I took biochemistry, the topic that we spent most of the quarter on, the first quarter of biochemistry, was intermediary metabolism. That is basically the functioning of the liver, okay? And it's just showing you some figures here on the rest of slide 12 and slide 13 of the red blood cell cycle, and you can see the liver there at 3 o'clock. on slide 13. It plays a crucial role in that. Okay, now remember that, and this is how it relates to the digestive system, that the red blood cells, they can only last about 4 months, and then macrophages in the spleen, bone marrow, and liver tear the old ones up and release the hemoglobin. The globin portion gets broken down into amino acids, and the vast majority of those are recycled. Not all of them, but most of them are. And then the heme groups are made in the various forms of bile. And we talked about this with the red blood cell cycle. Okay. And then, so on the slide with the gallbladder, on slide 14, it's showing you the various ducts that lead to the hepatopancreatic ampulla. And so the pancreatic enzymes, which is the primary source of digestive enzymes, are entering the small intestine at the very beginning together with bile. So this is why I'm talking about bile on slide 14. It is a complex mixture. We talked about bile salts in lab 3. There's a lot of other electrolytes in it besides bile salts, various pigments. And that's why bile, various forms of bilirubin are green and then kind of yellowish, orange, and it goes brown. But this is also the pathway of cholesterol excretion in your body. When your body needs to get rid of cholesterol, it'll put it in bile. The bile will go into the donut hole and you poop it out. Okay? So excretory pathway for heme groups from hemoglobin. That's what it says there for dead RBCs and hemoglobin. It's the heme groups. And then that bile salts, that is part of bile, will be the emulsifier for fat digestion. That is its role in the digestive system. Okay. Remember that bile is also a buffer, so you've got a bunch of buffers entering the very beginning of the duodenum, where the chyme from the stomach comes out really acidic, 1 to 2. Okay, but in lipid digestion, bile salts are emulsifiers, so we've covered that back in lab 3 as well as here. Slide 15 is showing you the various ducts, again, that deliver the bile. Bile is dripping out of the liver 24/7 because it is processing all the red blood cells that are taken up. Every day, millions of reticulocytes are going into circulation and millions of 4-month-old red blood cells are being destroyed. And so you're getting a constant dripping of bile from the liver. This is what the gallbladder is for, is you only need the bile to go out into the lumen when food is passing the pyloric sphincter. And when it's not, the hepatopancreatic sphincter contracts and the bile just backs up and goes into the gallbladder. This also, the slide is showing you why the biosalt is a good emulsifier. Remember I said an emulsifier is basically like soap, and soap molecules will have parts of it that are hydrophilic and parts of it that are hydrophobic, and that's why they can wash the skin oil off of your skin. The oil can mix with the hydrophobic portion of the soap, and then the hydrophilic portion of the soap will interact with the water coming out of the faucet. That'll rinse it off. Well, here you can see the biosalt is part hydrophobic, hydrophilic. We'll see that again in another figure that's coming up from your textbook. Okay, so we got the squirting out of the controlled release of chyme out of the pyloric sphincter. Those are tiny droplets. You got the movements of the small intestine, peristalsis and segmentation, that also are just constantly mixing. It's like constantly shaking the salad dressing bottle, oil and vinegar. And if you keep shaking it, you keep the bubble small. Yet still the hydrophobic fats want to separate into larger globules and layers. And what the bile salts do with these hydrophobic ends going up against the fat droplets and the hydrophilic ends up against the chyme, which is hydrophilic, you're able to keep small droplets small. That's number 3. You're keeping small droplets small. That increases the surface area for more lipase-driven reactions. Okay, and this is on the bottom right is showing you a picture of one of those tiny micelles. Slide 17 is actually showing you the bile salts and how it interacts with fat droplets, putting its fat-loving butt end up against the fat droplet, and then the hydrophilic ends are up against the chyme. That's on slide 17. And this is just, again, showing you what's happening that we talked about earlier with the absorption of fats. In that simple columnar epithelial cell, triglycerides are put back together. It seems kind of amazing, you know, why your body would break it up into 2 fatty acids and a monoglyceride just to put it back together. And it's because the triglyceride, even though it's fat soluble, is too big to get through the lipid bilayer. The only way it can get through is to break it down into a monoglyceride and 2 fatty acids. Then they can diffuse through the lipid bilayer. So then that simple columnar epithelial cell reformates it into triglycerides, put it into chylomicrons, and then spits it out by exocytosis. We're going to expand on this in just a few minutes here, but the triglycerides are not going to just stay triglycerides in the chylomicron. Once it gets into the lacteal, the chylomicron will break apart, and you will have these triglycerides now bind to various proteins. We can call these lacteal proteins, and since it's part lipid and part protein, we call it a lipoprotein. Now, this lacteal protein actually came from blood, because remember, lymph comes from blood. It leaks out of the blood capillary because all capillaries leak. It goes through it becomes interstitial fluid. Then the interstitial fluid is picked up by a lymph capillary, in this case, lacteal. It's going to go all the way in and go back into the blood here. So even though you'll see it highlighted on point number 5 as a lacteal protein, it is basically a plasma protein. It came from blood and it's headed back to blood. It just, for right now, is in the lacteal. And on point 6, it's showing you the various types of lipoproteins. Going back to the very beginning of the semester, and I talked about the phospholipid bilayer, the fluid mosaic model of plasma membranes. I called it a sea of protein boulders floating in a phospholipid bilayer. That was intentional even back then in the beginning of the semester. I want you to think of proteins as being dense like boulders. And now we can apply it to these lipoproteins. If you look at this figure on the bottom right and read point number 6, you will see that it is all shades of gray. How much lipid to protein ratio is it? Since boulders are dense, if you have a higher percentage of protein, that will be a high density lipoprotein. If you have a low level of protein and it's mostly fat, which is not dense, then you're going to have a low density lipoprotein. And low density lipoprotein, and they have a newer class called very low density lipoprotein. But when you have too much low density lipoprotein, it is very unhealthy. That is what is referred to as the bad cholesterol. We'll talk about this in a little bit more detail in a second. But the high density lipoproteins, what's oftentimes referred to as the good cholesterol, isn't good just because of the absence of bad. It's a actual protective type of lipoprotein, and the more of that you have in your body, the less chance you have of getting cardiovascular disease. And again, it's a protective effect above and beyond just the lack of bad. All right, here's some topics starting on slide 19 that we do have to cover because it's in the course outline. And that has to do with metabolic rate, specifically basal metabolic rate. Basal metabolic rate is the minimum amount of energy it takes to keep you alive. I don't need the whiteboard. Or cost of living. First bullet, minimal energy, and the organs that you need to keep you alive, and you can see that the two top tier organs are their brain and liver, but obviously your heart and your respiratory muscles to breathe, and your kidneys. So whatever those organs need with you doing nothing is basal metabolic rate. Now remember, rate is always something over time. In this case, it's hours, and it's kilocalories per hour. That's what that's the units of metabolism. They don't actually measure like how much ATP you're using. They are just going to go by how much oxygen you consume. And this is the third bullet. And this makes sense. If you are awake, fasted, and resting, none of your cells should be doing anaerobic respiration. All your cells, 100% of them, should be doing aerobic respiration. And so you can actually calculate how much ATP you're using up by how much oxygen you're using up. Okay, why do you think you have to be awake? You have to go back to the Lab 5 PowerPoint on sleep. If you fall asleep and you happen to go into REM, you are not at basal. Your body temperature is up, your heart rate is up, you got to stay awake. Fasting, you don't want to be having any smooth muscle doing peristalsis or segmentation. That burns up more energy necessary than just those organs to keep you alive. And you have to be still. Obviously, you don't want your skeletal muscles to be burning up ATP, so you have to be really still. So let's say we can calculate with aerobic cellular respiration that 1 liter of O2 is going to produce 4.8 kilocalories of energy. You just have someone awake, fasted, and laying or sitting really still. And over the course of an hour, they use 15.2 liters of oxygen. And then you just do the math, and that's the bottom bullet on slide 19. And that's a pretty normal adult basal metabolic rate in the low 70s. Now, it changes, and it's different for genders and for age. So even if you had a BMR at one point in your life, as you get older, it's different. This is relevant for why older people tend to get heavier. And we'll look at that in just a second. Males generally have a higher BMR than females. This is, of course, not including when females are pregnant or lactating. And also, this is a generalization that is slowly becoming obsolete. As more and more women are working out and getting in shape, the percentage of their body weight that's muscle is also increasing. And a woman who is really in shape and stays in shape can have a BMR very similar to males. That is because of their percent body weight that's skeletal muscle. Okay. And again, if you're eating and you're doing peristalsis and segmentation and secretion, your metabolic rate's going to increase. Also, one thing that wasn't listed on the previous slide, you got to be well. If you are fighting off an infection, whether it's localized or systemic, your body is burning more calories. All right, now let's tie BMR to TMR, total metabolic rate. And this chart that you see on the bottom right on the slide 20 is something that you sometimes see in the walls, pasted up on the walls of gyms that are all closed right now. And that's all has to do with calculating total metabolic rate. Total metabolic rate is BMR, how much calories it takes us to stay alive, plus the calories that you burn in certain activities. And that's why these things are in gyms. But this is like all day long. All day long, your BMR and your activity metabolic rate equals total metabolic rate. And this is basically the bottom line, balanced, healthy way of weight management. If you want to stay the same weight, you got to make sure that the calories in equals calories out. If you want to lose weight, you better make sure that there is more calories being burned than you're putting in. But if you're putting in more calories than you're burning, you're going to put on weight. And now let's go back up to the top of the slide where it talks about what happens with age. If you are eating the same and you're doing the same activity, because of decreasing BMR, your TMR is going to decrease and you're going to put on weight. So as you age, you got two choices. You either eat less or you exercise more. And if you don't, you're going to be the path of least resistance of putting on weight as you get older. Okay? Now, when I talk about this thing on the bottom of the slide, this is the balanced, healthy way of weight management. And that means something that you can do for the long haul, okay, your whole life. There are things that you can do that don't apply to those three bullets, but you better do that on the short term. And I'm talking about various diets that people can do that actually tricks the liver, and when the liver is tricked, you can lose weight. Since the liver is a top tier organ, I would not trick the liver for real long. Okay. Now I don't know what anybody's doing, and I don't know what all you guys heard, because there's so much misinformation about this, especially when people are salad shit. And one of the things why I'm glad that I didn't just go in my degree to be a dentist. I had to first get a bachelor's degree in biology, majored in biology, minor in chemistry and psychology. It gives me a more well-rounded understanding of science. And when I went to UCLA, one of the things that they really drilled into us, and I'm sure glad they did, was that science plus financial conflict of interest equals non-science. They can give you all the charts and graphs they want, but if they have a financial conflict of interest, if they're going to make money off of the interpretations of data and conclusions, that's non-science. If you have a financial stake in it, you can't speak about it. You need to have someone else who's independent, who won't make any money off of it, do the study, and then you can report that to make a buck. But if you're doing the study, when you're going to be the one making the buck, that's non-science. And so a lot of these diets that come out, will show you charts and graphs, and they're all done by either the company that they're of the product they're trying to sell, or something else it's not independent, that's not science. All right. There's a lot of things about keto diets. Keto diets work, but they are not the 3 bullets you see on the bottom of the slide. They are tricking your liver. They will work. Then maybe get it down, use it until you get to the point where your weight is where you're happy with. Then you better get to these 3 bullets for the long haul. The term keto diet is an old name. It's a new name for an old dog. And that old dog, one of the early keto diets was the Atkins diet. And that old dog died of heart disease because he did it for too dang long. And when you try to trick your body for too long, it's eventually gonna kill you. Okay, so 3 bullets is the balanced, healthy way, long term. All right, slide 21. Let's talk about the vitamins that, not all the vitamins, but a lot of the vitamins are absorbed in the large intestine. We can, a lot of times you will hear vitamins and minerals. The difference is vitamins have carbon, they're organic compounds, that's the first bullet in slide 21. Minerals don't have carbon, they're inorganic. We can take these vitamins and separate them into fat-soluble, hydrophobic, or water-soluble, hydrophilic. The 4 that are fat-soluble, just remember ADEK, A-D-E-K. And we've actually mentioned some of these over the course of the semester already. Vitamin A was the precursor to retinol. Vitamin K is the precursor to prothrombin, so I know we've mentioned those. Vitamin D, you need that to absorb calcium and phosphorus. Even if you have a good amount of calcium that you're taking in, if you don't have vitamin D, it just stays in the donut hole, never goes in. Vitamin E is, again, looks to be as helpful for the stability of cell membranes. Antioxidants, by the way, if you hear anything, again, this is another thing that's non-science. People who are selling things that are supposed to be good because they have antioxidants, it's bogus. Antioxidants in independent studies show to be about a wash. Half the studies show that it helps, half the studies show that it doesn't do squat. So when someone tries to tell you something because it has antioxidants, just roll your eyes at them because it's non-science. Then the rest of the vitamins are water-soluble vitamins on the next slide. Just a couple things to highlight off of this slide. What slide is this? Is this slide 22? Yeah, slide 22. I don't want you to memorize everything on slide 22 because that's what the internet is for. Just remember that vitamin B12, you need that for erythropoiesis. And if you don't have enough vitamin B12, that's where you get that pernicious anemia. We talked about that before. All these bullets on the slide, except for the very bottom diamond, are vitamin B complex, even though they don't say B on them. Those are all vitamin B complex vitamins. Vitamin C is ascorbic acid, and you need this for collagen and connective tissue, which is why if you have deficient vitamin C, you get scurvy. And the periodontal ligament, which is all collagen, gets weak, and that's why old sailors centuries ago, if they got scurvy, their teeth would fall out. Okay. Vitamin C, again, I've talked to you about, you can only absorb 60 milligrams. So make sure you remember the science, okay? All right, so we covered the organic vitamins. These are the inorganic minerals on slide 23. It shows you the ones that are really important. A lot of them are just the electrolytes that we need for nervous system, and muscle function. All right, and then the last thing on the digestive system is going to be about cholesterol. This is especially important in how it relates to health. Dietary cholesterol is not needed, nor do you use it to make ATP. You can, but you don't need to. Okay. Now it's got that classic steroid 4-ring structure that you can see on the bottom left, 6, 6, 6, and 5. It is included on all of your cell membranes. All steroid hormones start as cholesterol. And a lot of the cholesterol is also bound with lipoproteins, and it would be part of the lipid part of the lipoproteins. See, now look at, on this third bullet here on the very bottom of the text on slide 23, you see bound to plasma proteins, lipoproteins. On that previous slide, it was lacteal proteins. It's the same protein. Used to be in plasma, went into interstitial fluid, now it's in the lacteal. Eventually, it's going to get dumped right here into the left subclavian vein. It'll be a plasma protein again. It's the same protein. All right, 2 main types. And this is where I'm going to expand on LDLs and HDLs. LDLs are the bad cholesterol. And the reason why is that your endothelial cells have LDL receptors. And it can actually bind to the LDL and actually draw it into the cell, the epithelial cell, and then spit it out on the back end, on the other side, so that the LDL will actually be embedded inside the arterial wall. It's not laying on top of the arterial wall in the lumen, it's inside the arterial wall. And those are called atherosclerotic plaques, okay? And there's also, again, a very low density lipoprotein. Same thing. You got receptors for both of these on endothelial cells. High density lipoproteins, again, will have more protein. Just remember protein folders. And it isn't just the absence of bad, it actually does have a protective effect. Your endothelial cells do not have HDL receptors, and it tends to make the consistency of your plasma a little bit more fluid. Okay. Now, let's just, one other thing that a lot of people don't understand. For number one, your endothelial cells are everywhere. Because of that, you can get atherosclerotic plaques everywhere. But you tend to get an increased rate or speed of building up these plaques wherever the blood flow is most turbulent and the blood pressure is the highest. Now, when we go back to talking about blood flow, which is, we talked about this in terms of resistance. This is also relevant for these, talking about LDLs. But remember that at the ascending aorta, right on the outside of the aortic semilunar valve, is where you have the highest blood pressure in your body. Because even in the aortic arch, where you got 3 branches, the cumulative diameter is already increasing. By the time you get to your brachial artery, where you're going to say it's 120/80, actually in your ascending aorta, it is slightly higher, okay? Also, like the 3 branches that come off the aortic arch, those are nice smooth off-ramps. But if you go right above the aortic semilunar valve, the right and left coronary arteries come off at 90 degrees, which gives you a really turbulent blood flow, right where the blood pressure is the highest. So even though you can form atherosclerotic plaques anywhere, you tend to form them more in the coronary system because of that high blood pressure and the fact that it comes off at 90-degree angles, okay? But don't think that's the only place you can get it. You can get them anywhere. All right, 2 main sources of cholesterol. what you eat and what your liver makes. This is slide 25. Okay, again, what you want to get off of slide 25 is people say, again, who don't have any knowledge of science, don't eat that, it's got too much cholesterol in it. At which point you should roll your eyes and say, I don't care. Why? Because dietary cholesterol is not a problem. If you eat more dietary cholesterol, your liver will just make less. That's not the point. The point is what kind of fat do you consume? This is the second bullet. When you eat, as again, shades of gray, a higher ratio of saturated fats, not only will your liver make more cholesterol, you will excrete them into your bile less. If the ratio is more unsaturated fatty acids, and remember, and this we have to go back to lecture exam 1, this is about how many hydrogens are on this long chain. If you look at that figure on the bottom right, if it's saturated with hydrogens, they're all single bonds. That's a saturated fatty acid, it's all single bonds. If you put a double bond in the chain, you got to take 2 hydrogens off. That's unsaturated fats. If you have multiple double bonds, that's a polyunsaturated fats, and those tend to cause your liver to make less cholesterol and increase the amount of cholesterol excretion. Okay? So that is more important than whether or not you eat cholesterol. It's what kind of fats are you eating? And then the last topic on cholesterol has to do with this genetic condition referred to as familial hypercholesterolemia. That's a long word. The bad part about this is that this is autosomal dominant, which means you only have to have one of these genes and you got the problem. And if you look at the pedigree, the family tree on the bottom right of slide 26, you could always tell when it's dominant because it doesn't skip generations. Every generation is going to have it. All right. The liver also has LDL receptors, but you want the liver to have these because what the liver is doing is it's taking the LDLs out of circulation and it brings it in by receptor-mediated endocytosis. When you have this condition, and you only need one of them because it's dominant, you're going to have way fewer LDL receptors in your liver, but you will still have LDL receptors out on your endothelial cells. Because your liver cannot take out LDLs, the LDL counts get abnormally, freakishly high. Okay? Now for the LDLs to form atherosclerotic plaques, the LDLs have to become oxidized, and that's why people have thought antioxidants could counter this. And as it says, the jury is still out. Whenever there's a study that says, oh yeah, this really helped people with this condition, another study will come out and say, well, it really didn't really help. Hopefully it will help. Okay, any questions on digestive physiology before we go moving on to the next organ system? No questions. Are you guys there?
We're here.
Are you watching something else?
We're here.
Did you go on a walk? You just left your thing on with your name up without cameras. By the way, there is increasing chance that emergency remote teaching, ERT, will most likely be here in the fall. Summer is ERT, and Cal Poly has already said that their fall is going to be ERT. I think, again, it's not definite, but the handwriting on the wall I'm seeing is that it's probably going to be like this in the fall. If you are taking classes, planning on taking classes in the fall, online exams are no longer going to be unproctored. Everybody in the sciences anyway are going to have to take tests on camera. And you can't just put your name up there. You're going to have to be on camera to take tests. So you're going to have the equipment. You're going to have the high-speed internet access so that you could take a test and still have your video up the whole time. Just a heads up, because I've had my own experience, but talking with other teachers here, it has been extremely disappointing, especially for those that are planning to go into healthcare, how deficient our students are in ethics. The cheating has been rampant. Chemistry professor wanted to do one more year, and with only 1 month's notice, he said, "I'm out, I quit." And one of the things, it's multiple things, but one of the things that really bummed him out was students cheating without any reservations. So classes in the fall, if we have to do this, you're not going to be able to take tests without being on camera. And it's programs that are watching your eyes. So if you're looking at something other than the screen that you're taking the test or your keyboard, it's already red flashing. Okay? And that's, I mean, it's ridiculous. It's ridiculous to cheat in a prereq class, because what are you going to do with a bogus grade? I mean, what are you going to do? And I know that there's cheating in nursing programs. Why? Why? You think you're going to be good at your job if you've cheated to get through? You think you're going to have a chance of passing the NCLEX if you've cheated your way through? That's beyond stupid. All right, let's go on to the urinary system. This actually I forget how many slides here. Way back when I first applied for this job, I had been teaching at Cuesta for 4 years, part-time. And when I applied, I had to do a teaching demonstration. And these slides, they work. So I put it in this PowerPoint because we had to do urinary physiology as our teaching demonstration. This picture on the top right, slide 27, is actually a slide of an album cover. It's a British rock band way back when called The Who. The title of the album is Who's Next. They were supposed to look really cool next to this concrete monolith, and all he did was take a piss on it. All right. Again, in order to understand urinary physiology, you do have to understand urinary anatomy. We're going to cover some basic functions and the sequence of those functions. Eventually, we'll be talking about regulating urinary physiology with hormones. Okay, slide 29. This is really important to understand. The primary function of your urinary system isn't to make pee. That's not the primary function. The primary function of the urinary system is to monitor and maintain body fluid levels, not just how much fluid, but what's in them. So it's volume, concentrations, pH. This organ system is about homeostasis. That's the primary function of the urinary system. And this, I think, is the last time you have to see this slide on the bottom of slide 29. Again, it's about water. And our whole body is about 60% weight is water. 2/3 are ICF, 1/3 is ECF. ECF is split up between blood plasma and lymph and interstitial fluid. The urinary system is monitoring and maintaining ECF, and eventually ECF has its influences on ICF because of transport processes. And in order to maintain ECF, to accomplish that, your urinary system as a secondary, oh, by the way function, has to get rid of certain chemicals that are waste products. Urea, ammonia, uric acid, creatinine, got to get rid of them. All right, really quickly, slide 31. This is one of the simpler organ systems, has the fewest organs in it. 2 kidneys, 2 ureters, 1 urinary bladder, 1 urethra. That's it. That's all there is to this whole organ system. And slide 32 showing a picture from your textbook. Then when we look at a kidney from the outside in, we have a fibrous capsule that separates the kidney from the posterior abdominal wall. Remember that the kidneys are in the posterior abdominal wall. They are not in the abdominal cavity. The outer portion of the kidney is called the renal cortex. This is where the renal corpuscles are. And then the inner portion of the kidney is the renal medulla. This is where we have the nephron tubules, the vast majority of nephron tubules. There's a little bit in the cortex, but the vast majority of it is in the medulla. Now, highlighted underneath this is something that's really key, something we'll be talking about on Wednesday. And you didn't have to worry about this in anatomy, but you need to know this for physiology. The deeper you go into the kidney, the more progressively salty it gets. The interstitial salt concentration is getting more and more concentrated the deeper you go in. The very center of the kidney is all the minor, major calyces and the renal pelvis. The renal pelvis forms a funnel that seamlessly transitions into the ureter. Slide 34 is showing you some of these structures of the kidney. Now on that slide 34, I want you to pay attention to the upper right portion of that textbook figure. All the nephrons, hopefully you remember that term nephron, begins in the renal cortex, because the renal corpuscle is where it begins. Now look at that figure. All the tubules and the nephron loops are in the renal medulla. And the last portion of the nephron is the collecting duct, and the collecting duct ends at the renal papilla. That's where all the nephrons end, renal papilla. And then that fluid drips out of those little holes into the minor calyx. Now, if you look at that figure on slide 34 on the upper right, the fluid in those yellow tubes, your body can still change. If your body can still change the fluid, it's called filtrate. As soon as it falls off those little holes into the minor calyx, your body can't change that fluid anymore. And the name of the fluid changes to urine. And slide 35 is showing as an introductory slide on the nephron. The nephron is the functional unit of the kidney. Each kidney has anywhere between 1.1 million, 1.2 million of these nephrons. And the bottom left shows you from start to finish. In green, where the Bowman's capsule and glomerulus, those together is the renal corpuscle. That's the beginning. It's out in the cortex. Then we're going to go through the various tubes, and it ends in the collecting duct in yellow. And notice where the bottom of the collecting duct in yellow is. That's the renal papilla. That's where all the nephrons end. Okay? So we'll start on slide 36 on Wednesday. Any questions? Let's take a second look at the whiteboard, the grade scale. Bacterial counts are in brown. We talked about the stomach last week, so I just started this on the small intestine. Again, purple is mechanical processing. Blue is secretion. Red is digestion, which ends in the small intestine. Green is absorption. And then we have the little schematic of the absorption mechanisms for that table. The 1, 2, 3, and 4 are the columns of that table. Any questions?
No questions.
Okie dokie. See you Wednesday.
Thank you.